Hello, students. Speaking live to you from McLean High School, I am Mr. Bouchard, one of your biology teachers, along with the lovely Miss Murdoch, who is behind the camera.
I'm here.
Today we're going to do the Algae Lab. And we would usually be doing this in person, but since we can't be in person, we're going to do this for you. And let's talk a little about algae first. Algae, you're like, what is algae? Well, algae is a unicellular photosynthetic organism. They're living, and they're in this water right here. That is what's making this green. I can't even think of how many fathoms.
So that's not food coloring?
No, don't be ridiculous. This is full of microscopic organisms making this water green. And what we've done back here is we've taken a little of our own and we've diluted it a little. All right, so we still have them in here, just diluted it a little. Now, you're like, what are we gonna do, Mr. Richard? We have another solution here. And this has water with nitrogen. And you're like, what does nitrogen do? Well, nitrogen is a molecule that helps supply, it's in fertilizer. And you're thinking to yourself, fertilizer, why have I heard that before? Mom and dad put that on the grass, or I've seen that this. And what fertilizer does is help with growth and provides nutrients to these little organisms here. And sometimes we see this happen in runoff as well. And when you see mowing lawns and water runs, and sometimes will run off some of that fertilizer into the gutters. And down the gutters these go, down to the, what are those called, Miss Murdoch?
Lakes and ponds and streams.
Ponds and streams. I knew it was something fancy. Lakes and ponds and streams. And then when that fertilizer gets there, it then spurs growth from these guys. So what we are going to do today...
Is that bad if they grow too much in a lake or a pond? Is that bad for the environment?
It can be bad because it can suffocate fish. It can not allow light to get through because even aquatic plants need light. And if there's a blanket, think about it. It's dark and there's plants like, I can't see a light. Or a plant, or the fish like, I can't breathe. Because this is taking everything and it just destroys them. It's very bad. It's called overgrowth.
Eutrophication.
Eutrophication. I knew it was a fancy word. Eutrophication. Try saying
a word. Yes. That's a
and
word.
You sometimes would see this they talk about this in the Potomac River a lot and they have things called riparian buffers, which you may be aware of, which you would see big growth on the sides of the water that sort of prevent pollution and stuff from getting into the water, i.e. Fertilizers and other growths, but it can't protect it all. And when you take that away, it just runs right off into the water.
So when you have lots of sidewalks and parking lots and asphalt, slick surfaces without plants and roots and things in them, and it's right next to the water, and then people have their cars and their lawns that they're fertilizing with a lot of nitrogen. It runs into the water much faster than if you had lots of plants and trees there to stop that runoff. Exactly. So I think that's what Mr. Bouchard was explaining. That was a really good point.
Oh, thank you, Ms.
That's why you want to keep your plants and things next to the rivers. It protects them from too much nitrogen runoff.
Excellent. So what we're going to do today is we're going to test the growth of algae. And what we have is we have our nitrogen solution right here. And we are going to put different amounts of nitrogen into different cups. Now, each cup, I'll demonstrate, is going to have 100 milliliters of just plain old algae water that's been diluted. And we've filled up all these already from this. So they've all come from the same source. So I'm going to measure out 100. And I've done this already for all of them.
So are you looking at that meniscus, Mr. Bouchard?
Well, I haven't gotten down there yet. So picky. Give me a second. I got a little bit down there.
He's being very careful to make everything exactly 100 mils. That's good. There is a dropper right here. Here, you can use this dropper if you need to move.
Thank you, wise sage advice.
I was ready for you.
Wise one. There we go. Excellent. Good. So there is 100 milliliters.
Good.
Looking good. Of the pond water or algae water. Sorry.
Well, it could have been from pond water, but it actually isn't. It's from that green tank over there by my window. That bright green tank that has all the algae in it. And we are testing it. We are using the algae as our test subjects.
So what I have done here is I have 2 sets of cups, and I've labeled it 0 milliliters of solution.
Nitrogen.
Nitrogen,
So
sorry.
there's no nitrogen in that one, but there is algae. Okay.
There's algae. This one is going to be 5 milliliters.
So 5 milliliters of nitrogen.
And then this one's going to be 10 milliliters.
So 10 mils of nitrogen.
And then this one's going to be 15 mils of nitrogen.
Okay.
And so what I have done is I will have my little baby graduated cylinder. He's only graduated preschool. This guy over here has graduated high school. That's why he's bigger.
That's going to be more precise for 15 mils than this
yeah.
other one, which is just 100 mils, which is going to be less precise if you're trying to measure a smaller amount of liquid, which is just good science.
Yeah, so let me go down. Sorry, this takes a second.
So it looks like you have a set of cups here that's already done.
Yes, I've already done one set.
0, 5, 10. Okay, and when we're all done, we're going to have
So I've done that
eight
at
cups here.
I've
Excellent.
done this at 5.
Okay.
And I'm now going to pour this into my 5. And so that I don't make sure I pour anything into it again, I'm going to move it up here away from me, as well as it did the 0.
Okay, all right.
Then I'm going to have to do the 10. This takes a little bit, guys. I'm sorry.
That's all right.
But you...
So here's a question. Is this going to be something we're going to see right away? Are we going to see growth of the algae right away and we're going to get to measure it? Or is it going to take longer than that?
Are you asking me, Miss Murdoch, or this is a hypothetical for the video?
No, I'm asking you. Is this going to be a test an experiment that goes over a long time, or is it going to be just right now today?
Oh, no, don't be ridiculous. This takes a while. You just don't add fertilizer and watch a plant out of the ground. No, it takes time. So what we're going to do is we're going to measure this every day for probably about 2 weeks. 2 weeks, would you want to do, Miss Murdoch?
2 weeks is usually what what it takes. We've done this for many years now, actually.
10.
And so the 10 mils of nitrogen is going into the 10 milliliter cup with that
I've
algae.
labeled them 10.
There we go.
Now the next one's 15. So yeah, we're going to do this over 2 weeks, and we'll talk to you. We'll take the measurements for you guys, and maybe if somebody wants to remind us occasionally, like, hey, Mr. Bouchard, did you measure? You know, because, you know, sometimes I might forget, and we got to make sure we get those. I probably won't forget. I'm like an elephant. Or a dump truck. Miss Mara called me a dump truck once. It was a very sensitive time. Just putting this in there, guys. This takes a second. Sorry, I know, but hey, you get to look at my lovely face while I'm doing this. Congratulations. It's a win-win for everybody. Well, you know, we got to see a lovely video of Mr. Bouchard today.
So we have 0 mils, 0 mils.
This is
5 mils, 5 mils.
10
mils, 10 mils. And 15 mils and another 15 mils coming up, and that is the set.
And that's
Those are
10.
going to go into the window and be in the light. And as the algae grows, we would expect that the water would become greener and greener as there are more and more cells in there. And each cell has chlorophyll in it because they photosynthesize. So it should, in order to see the growth, we should get water that looks greener and greener. And in the next video, we're going to explain how we're going to measure that.
And then I had to refill this one once halfway with 5 because it was only a 10. So I measured a 10, put 10 in. Now I'm putting 5 more. So there I have my 15. So again, I have my 15, 10, 5, and 0. And I want you to really think about what you think that 0 was there for.
And why there are 2 sets of cups and what that means. Yes.
All right, guys.
And what the IV and the DV would be for this. You need to think about all those things too, and what the constants are for this experiment, besides Mr. Bouchard's beautiful face.
Again, guys,
A very
thank you.
important constant.
We'll be on. Bye.